Hello guys, hope everyone is doing well. Today we'll be starting with IBMYP biology syllabus, and we'll be starting with unit 1, which is how is life organized, and it talks all about cells, cell theory and stuff like that. So today we'll be focusing on cells, the cell theory, the characteristics of life, and something interesting, if viruses are alive or not, and then we'll be looking at Louis Pasteur's experiment. To begin, we need to look at the cell theory. Okay, so so before, in the 1700s and something like that, biology was just observations, but we always thought that there must be some underlying feature, something really small that must be stimulating all the things that we do. So with the development of microscopes around in the 1800s and the 1900s, we developed the cell theory. We found out that the cells are the basic building blocks of structure in living things, which means that cells are the smallest unit of life. Cells themselves are living, and cells are made from other cells by division. Okay, so cells are the smallest unit of life in the sense that if you take one cell, a single multi-cell, sorry, a single cell, a unicell, then what would happen? And put it, if you cultivate it outside, or if you subject it to its thriving conditions, it would grow, it would survive. However, if you took something smaller than a cell, which are the cell organelles, they wouldn't survive. That means cell organelles are not living. Cell is living. Therefore, the cell is the smallest unit of life. And cells are the building blocks of structure in living things. So cells come together, form tissues, then tissues form organs, organs form the organ system, and organ system the whole body. That's how classification and the development and the building blocks of living organisms is. Multicellular organisms. And cells are made from other cells by division. So if I have one cell, this cell would then divide, and this is known as mitosis. So there are two ways cell division can happen: mitosis and meiosis, but we'll focus more on it later. It comes in a later topic. But how do we define something as living? How do we know something is living? Well, they should exhibit seven to eight characteristics of life, and instead of this, just focus on Miss Gren. For MYP, this is a short form or an abbreviation we would look forward to, where M is movement, R is response, G is growth, sorry, S, there's I missed an S. S is um S is sense. You can just call it sensory. So it senses its surroundings. Then Miss Crane, what else is left? R, you can call that reproduction. Okay, you can call R as reproduction, and you have E, which is excretion, and N, which is nutrition. Okay, so E is excretion, and N is nutrition. Sorry, my handwriting is a bit bad. Nutrition. Okay, just look them up here. So nutrition means that you obtain nutrients by feeding or making food. That means you basically consume food, ingest food, and then you generate energy from, you break the food down to release energy. And every living thing does that, every living organism. Without it, no living organism would be surviving. Respiration is the breaking down of glucose to release energy. So when you ingest food, you respire. So you break down food into glucose and then you break down glucose to release that energy. And every living organism has to respire. Then you have to grow. That means achieve a permanent change in size or mass. Growth can also mean size is increasing, your mass is increasing. So when cells divide, they split like this. So at this point their size has increased. Then is excretion, to get rid of waste products by chemical reactions in cells. So if it's a multicellular organism, you would have your excretory organs such as your anus and stuff and organs like that. But in cells, it just happens through the cell membrane, where the cell membrane regulates what goes in and out of the cell, and you just excrete from there in unicellular organisms. Then homeostasis is to maintain a constant and stable internal environment. You need to match the temperature of your environment and your inside. Reproduction is to produce offspring. Even cells reproduce. This parent cell would have a daughter cell, and that's through cell division. And movement is to move from one place to another. You may be wondering, plants don't move, but plants do exhibit tropism, and that is also another chapter. Plant tropisms. So sometimes plants move in the direction of sunlight, exhibiting phototropism. Yeah. Okay. So there are certain exceptions, but this is a general overview of how do we call something living. What is the difference between a living thing and a non-living thing? Now let's look at viruses. Well, viruses are basically a free-flowing genetic material which are basically dead without a host cell. So viruses are genetic material which have a protein coat and a free-flowing RNA. Okay, RNA or RNA. So viruses have RNA and they inject that into the human cell where it becomes a viral DNA. That RNA in the human cell becomes a viral DNA. And viruses are non-living because they do not conform to all the 7 characteristics of life. For example, without a host cell, can viruses reproduce? No. Can viruses move? No. Do you think viruses excrete? They do not excrete. They require a host to reproduce and synthesize products of metabolism. So without a host cell, they are basically dead. Inside a host cell, they may exhibit some characteristics of life, but in most of the cases, they do not. And then you have Pasteur's experiment. So this just acts as an evidence to cell theory. One of the features of the cell theory that says that all cells come from pre-existing cells through cell division. Yes. So what Pasteur did, so he took two beakers. He first sterilized them. Sterilized means he killed all the prior bacteria and microbial growth that was present. He then closed them with a sterile flask. In one of them, he put this swan beaker, but in the other, he did not. Okay, so one had the swan necks nabbed off, whereas the other remained intact to prevent the entry of unwanted microbes. So this acted as the control group, whereas this acted as the experimental group. The flask without the swan neck turned cloudy. So it turned cloudy. So he had some broth filled up in these flasks, and this broth turned cloudy because of microbial growth. The flask with the swan neck remained clear. Now, before, people used to think that microbes and things can just spontaneously appear. For example, if you have a dead rat, yes, you have a rat that is dead, then mice and not mice, sorry, microbes just appear on it. It starts to degrade, it starts to decompose. And people used to think that this just happened. But then this experiment proved that once it's exposed to air, there are free flowing microbes. So the viruses, bacteria, they're free flowing. Yes, and all of them come here, and then so because of these and its exposure, they start dividing here, because this provides the ideal growth conditions, ideal surviving conditions for these microbes, and then they start reproducing. So it's not spontaneous, it's reproduction. So this was it, but I would like to ask you guys a question, which would, wait just a minute, which would make you question the cell theory. It says that cells are made from other cells by division. Well. If so, what about the first cell? If cells are made from other cells by division, what about the first cell? How did the first cell come to be? This first cell does not follow this, it's an exception to this theory. However, how did it form? Well, you guys should research on this, but this will be covered into your TP years in grade 11, and this is not covered in MYP. However, just think about this question. Okay? Thank you, guys.